So guys, to continue our series on conquering the final frontier, we're going to talk about Mars, and more specifically, how we get all the conditions we need to survive up there. Our guest today, Dr. Benson, is an astrobiologist currently doing her postdoc research at the 6th floor of Byhall, and has come to talk to us about some exciting plans of hers. In 2 months, Annie is going to Mars to assemble and stock some terrariums with various microbes. The goal? To see how some of Earth's most hardy, useful, and unique microbes fare in the much more hostile Martian conditions. The finding of this decade-long project could have huge implications for future energy generation and the habitability of our potential second home. So, Annie, before you leave us for the cold mountains of the Utopia Planitia Basin, do you care to tell us about a few of the coolest microbes you're planning to put in your suitcase?
Of course, and thank you so much for having me. Where would you like to start?
So I've always been fascinated with the issue of radiation in space, and places like Mars that lack a significant atmosphere for protection. Are you planning on working with any small boys that would actually be able to survive that?
Yeah, it was not easy to find microbes that seemed capable of withstanding the extreme radiation found on the planet.
Yeah, isn't spending 180 days on Mars equivalent to getting something like 24 CAT scans?
That is exactly right. And to think growing up, my mom was worried about the radiation I was exposed to from talking on the phone for too long.
And now you're going to be on a planet with an atmosphere 100 times thinner than Earth.
Well, at least I'll be wearing a spacesuit, but my coworkers and I have not yet mastered the art of making spacesuits for our little microbial friends. So instead, we had to find one with a built-in radiation suit. The bacteria I'm bringing up there is called Deinococcus radiodurans, more commonly known as Conan the bacterium, because it's nearly indestructible. My lab will be examining it to determine potential effects of radiation on living organisms.
How do you know this meager little pal will be so suited to living in such a dangerous environment? Like, where is it found on Earth?
The first time actually this microbe was observed, it was in a tin of canned meat, spoiled from being x-rayed too many times. And it's pretty easy to culture.
Whoa. Yeah, so I guess if you're saying that if we can grow it in canned meat, it could also be grown in the lab.
Yes, and it's impressively resistant to ionizing radiation, UV light, hydrogen peroxide, and so many other things known to damage DNA. It also doesn't need much moisture at all to avoid desiccation.
Desiccation being drying out, of course. So, where do you find these bitsy beasts?
Well, only 7 strains in total have been found on Earth. Some in hospitals, llama feces, in hot springs, haddock tissue, of all places. Based on our 16S rRNA sequence analysis, my lab determined that Deinococcus line of microbes has several totally unique proteins.
For those of you who don't know, 16S ribosomal DNA is basically a part of bacterial DNA that all microbes have, but within each group of microbes, the DNA section has a few differences. This shared section of DNA is perfect for grouping microbes, since we can group them based on those little differences in their 16S ribosomal DNA strands.
Yeah, that's exactly right. And in the Deinococcus microbes, genes that code for creating proteins linked to DNA repair and environmental stress response seem to have been horizontally transferred from eukaryotes, so aren't found in most other bacteria.
So basically, these modest fellas have evolved to be able to make proteins that no little fellas can make, and it's led them to be able to handle living in some truly absurd environments.
So cool, right? And it's also worth mentioning that they're talented at repairing their own DNA once it's damaged too.
Damn. So, what do you think are some implications for these abilities? What could be some uses for these adaptations?
Well, on Earth, they're currently being used to treat mixed radioactive wastes that contain ionic mercury and other toxic metals. They can detoxify or immobilize a variety of toxic pollutants that are byproducts of some of our most efficient ways of generating energy. They're also generally convenient to send into places where humans don't really want to send themselves. That's why I think it'll be an excellent specimen to study on Mars. By looking at how they react to pretty extreme amounts of radiation found on Mars, extreme by Earth standards at least, we can hopefully gain some insight into both if and how it's possible for cells to survive there.
Incredible. So, this bacteria will hopefully indicate ways that earthly life can survive the harsh radiation of Mars. Well, with implications that awesome, how could any other organism follow this teeny trifler?
Well, let me introduce you to a few microbes just recently discovered at the Willerslev lab at the University of Copenhagen. The research team there took ice and soil samples from permafrost sites in northwestern Canada, northeastern Siberia, and Antarctica. By extracting DNA, they discovered that some of the organisms living there are more than 500,000 years old.
Whoa. I'm blown away. I mean, how do they even do that? From what I know of DNA extraction, many methods can't tell the difference between living and dead cells, since DNA is DNA no matter what. So, could they have been looking at cells that lived and died 500,000 years ago and have just been preserved?
That was a huge concern, and the researchers were hyper aware of it when conducting the experiments. They also had to deal with the presence of organisms that were technically alive but metabolically inactive.
So they really aimed to target only the wee bitties that were still kicking, and vigorously.
The key to differentiating active and inactive microbes is in the DNA. DNA from active microbes is generally not as degraded as the DNA in microbes with active metabolisms. All cells decompose and break down over time, but some are better than others at repairing that damage.
To do that, they'd have to use energy, so they'd have to have active metabolisms.
Yeah, of course. And parsing out the dead microbes was also a challenge, because cells can stay intact long after they die. The researchers decided to only look at DNA they collected that had more than 4,000 base pairs, or like DNA segments, because fossil records have shown that DNA from dead microbes would probably have broken down over time to only exist in really, really short segments, much shorter than 4,000. So they really limited the volume of data they could use in order to make the data as accurate as possible.
That's amazing. So, what we're saying is we can really trust that the wee beasties are in fact 500,000 years old.
Yeah.
So, what are you hoping to learn from them when you get them up there in Mars?
Basically, we know that they can survive and thrive even in very cold environments, and that they've been around for a very, very long time, which leads us to believe that they could be strong colonizers in a new environment. With these, we're just hoping to stick them in a terrarium on Mars, exposed to the natural conditions there, and see what they do.
Well, I, for one, am fascinated with what you might find. And we're approaching the end of our time together today, so why don't you tell me about just one more?
So, one of the challenges we face up there is limited access to nutrients. Living cells tend to rely on other organisms' participation in nutrient cycles to get the food they need. Since, after all, once an organism uses up, say, ammonium, which contains nitrogen, the byproduct they leave is nitrite. Then it's up to another organism to use that nitrite and turn it into nitrate, etc., until it goes through a cycle, and it's again at the ammonium form that the first organism can use.
Okay, so are one of these cramped critters able to use, say, nitrogen in any of its forms in the cycle? So it wouldn't require a community effort?
No, that would be cool too, though. But instead, we found a microbe that has such an easy to generate energy source that we can just keep making it and never need to change it back into its original form. It eats electricity.
Whoa.
So basically, the way this microbe works is that it's a member of a group of microbes called lithoautotrophs, or they're rock eaters, basically. This microbe will take some inorganic substance such as sulfur, magnesium, iron, which is a whole lot of on our red planet, and use those elements to assist in the metabolism, I guess you'd call it, of electrons in its environment. This microbe needs three things: iron, water, and electricity. With solar panels, we can get one. The whole planet is basically made of another, and there are rivers hidden just below the surface of Mars that could easily provide the last necessary step.
First off, that's incredible. But how do they physically do it? How do you eat electricity?
It's a process driven by a single unique enzyme. Once Methanococcus excretes it, it hangs out on the surface of an electrode where it takes up an electron. It also snags a proton from water, then it can form a hydrogen atom. And hydrogen is a major food source for some cells.
You mentioned earlier it can do something else exciting. Something about methane?
Oh, yeah. The other thing about these microbes is that some of them are electromethanogens, which means they convert electricity into methane. You know, cow farts.
Well, that's time I checked, farmers were trying to use cow farts to power their tractors.
Yep, and that's exactly what we want to do. Use bacteria farts to power our Mars tractors. So some of these microbes are known to produce methane with 80% energy efficiency. That's really, really good. These results show that methanogenesis can be used to convert electrical current into a biofuel. This has huge implications for studies on Mars. Biofuels are an energy source that we understand pretty well here on Earth. And if we can use these guys to synthesize, say, methane, then we'll have one of the pieces of a combustible fuel source. I'm not saying rocket fuel is made only of methane, but it is certainly a key ingredient.
I want to move to Mars.
Not yet, you don't.
Okay, but from the past 10 minutes, you've gotten me really excited about the future of humans on Mars. And regardless of my ability to live there, you should keep us all in the loop about everything that you and these small wigglers are up to.
Of course, and if you're lucky, maybe I'll bring you back some space aliens to talk in the next podcast.
Well, thank you guys all so much for listening. I hope these past few minutes have gotten you excited about the seemingly unquenchable human desire to dominate any surface we can get our hands on. This was the latest edition in our Conquering the Final Frontier series, and as always, our sources are linked in the description. Next week, our guest Dr. Bacchus will explain to us how Chlamydomonas is the currency of the future. I'm Nico with our guest Dr. Annie Benson signing off. May you all live long and prosper.